my long-term interest has been cancer research, and I had some experience in the field even before I took my degree, but from a physics perspective, radiation physics perspective. Then I moved into being interested in the effects of radiation on mammalian cells, with the practical linkage being to the treatment of cancer with ionizing radiation. So that was the theme that I was pursuing. And the Ontario Cancer Institute was something different in Canada at that time, because the major effort was around assembling a research group that was multidisciplinary, that represented a range of biological disciplines, plus some areas of expertise in physics that were relevant to cancer. One of the early efforts was to help people to get to know each other. And so there were evening sessions for this newly recruited staff to meet each other and learn about each other's research interests. And that's where I met Dr. McCulloch, who had joined the staff of the Biological Research Division, whereas I was on the staff of the Physics Division. The kinds of experiments Dr. McCulloch was interested in were studying the influence of transplants of bone marrow on mice that had received large doses of total body radiation. It had already been shown previously that if mice given large doses of total body radiation received no bone marrow transplant, they wouldn't survive. But if enough bone marrow cells were transplanted intravenously, they would survive. And he wanted to do the same kinds of experiments, but wanted to, for reasons that I don't remember too well, wanted to do quantitative work. What happened if you gave different numbers of bone marrow cells? And there had been actually very little work of that kind done so far. But to irradiate the mice, he needed to use the equipment of the physics division. And the head of the physics division, Dr. Harold Johns, who was my boss at the time, wanted somebody to help him, help Dr. McCulloch with his experiments, so that the radiation part of it would be handled by somebody who was well qualified. So that's how our collaboration got started. And very quickly we discovered we worked together very well and in complementary ways. And we very quickly ceased to be specialized members of a collaboration and became a genuine cooperative pair who discussed everything and came to decisions on the basis of discussion and careful thought. In terms of actually doing the experiments, we again each did just about everything. So I learned the biological techniques, and Dr. McCulloch became quite conversant with the relevant physics. And the trust that we built up at that time, I think, was crucial to our subsequent work. We were lucky that we put the right number of marrow cells into these irradiated animals so that there were discrete lumps, and they did turn out to be colonies. And in fact, they did turn out to be clones. But they were there in numbers small enough that we could see them as distinct bumps. And Dr. McCulloch didn't overlook them. They could have easily been overlooked, but he was doing a thorough job, and he noticed them. Now, we weren't the first people to actually see them, we found subsequently. I mean, we didn't know that at the time. But the interpretation that had been given to them was that they were local areas of regeneration, which to me implies that multiple numbers of cells were probably responsible for these local areas of regeneration. We interpreted it differently as colonies, individual colonies that might have been derived from individual cells. So we were lucky again, and perhaps the luck was because we had prepared minds. We were quite, because of our background in cell culture, both of us had that kind of background. We didn't think about local areas of regeneration. We immediately thought about colonies. And so we didn't misinterpret the results. And I think another thing that turned out to be fortunate is we tried very hard not to overinterpret the results. In other words, all we knew about these cells was that they came from bone marrow and that they could give rise to these bumps, these colonies. We didn't know what they were. We, of course, soon began to wonder if they might be the stem cells that had been talked about for 50 years or more previously. The concept had been around that time, but nobody had been able to identify these elusive stem cells, and the speculation had been mostly about what they might look like. Well, it was well known that they would be a small minority of the total number of cells in bone marrow, and it was very likely that they would be rather featureless. I mean, they would have no specialized characteristics because they're the source of the specialized cells. Some very careful experiments were done by a member of the group who was actually a graduate student at the time, who came from a medical background. Dr. Andy Becker did some really rather challenging and very pretty experiments using radiation-induced chromosomal markers to demonstrate that, in fact, the colonies in the spleen were clones derived from individual cells. And at that point, we knew we were dealing with cells of great interest. And we then began to wonder more seriously if they were, in fact, these elusive stem cells that have been talked about for many years. And we faced the problem that there was no accepted definition for stem cells at that time. And so we had to develop one. And we identified some properties that we thought were essential for characterizing these cells. One, that they'd be capable of extensive proliferation. Another, that they be capable of self-renewal, a crucial property, because if the stem cells are going to persist and be able to regenerate the blood-firing tissue, they have to be able to produce more cells like themselves. And of course, they must be able to give rise to differentiated cells. And the challenge was to study their properties. I mean, an analogy I heard used is, suppose you have a package of seeds that you want to use in your garden, but the package happens to be a bit old, and so a lot of the seeds aren't alive. But when you look at them, they all look the same. And the only way you can tell which ones are alive is by planting them and seeing what grows. And the analogy would be those seeds that can grow to give rise to a complete plant would be the equivalent of stem cells, and those that couldn't multiply at all or could only multiply a few times and give rise to the beginnings of a plant would not qualify as stem cells. The crucial properties of colony-forming cells that made us think that they were stem cells included ability to undergo extensive proliferation, the ability to give rise to more than one kind of specialized blood cell, and the ability to self-renewal. And the experiments that were carried out to test for ability to undergo self-renewal involved dissecting individual colonies from an intact spleen, so the cells were still living, dispersing the colonies, and then transplanting those cell suspensions derived from individual colonies back into irradiated mice to test them for their ability to give rise to new colonies. These experiments were initiated by Dr. Illusive, who had joined our group and in fact had been a senior member of the research staff at the Ontario Cancer Institute from the beginning. He was instrumental in the thinking that led to the definition of stem cells that we arrived at. The surprising result that we obtained was not that individual colonies could give rise to new colony-forming cells, but that the distribution of new colony-forming cells per colony was very heterogeneous. Small minority